one that kind of looks like this, and another one that looks like that. And those would be labeled C1A and C1B. So when they get split, you add a letter to them. Right? Is that a C? It's a 1. It's a 1. Those are all 1s.
C1.
Yeah, C1. So they're all numbered. C1, C2, C3, however onward. So we're going to go at 2 parts to the complement protein, the complement system. We're talking about how they recognize pathogen and then how they might respond to pathogen. Because in both cases, there are really 3 different ways. Activation. There are 3 different ways complement protein is activated or split. Right? Number 1 is what we call the classical pathway, because it was the first one discovered. And it's probably the easiest one to understand. Here's our pathogen, right? Our nice bacteria. He's got some antibodies hooked to him. If there are enough antibodies clustered together, that's where C1 is going to bind. It'll bind to the arms of those antibodies. And that's what's going to cause it to split to C1A and C1B. Okay. The classical pathway is just complement C1 binds to antibodies. And there has to be a bunch of antibodies clumped together, which is why this usually happens on the surface of a pathogen. The alternative pathway is the hardest one to understand. As I just told you, complement are numbered. There are some supporting molecules that are lettered. I don't know why somebody did this, but if 3 of those supporting molecules all bind the same pathogen, so if molecule B, D, and P all bind that same pathogen, instead of C1, what these really do is these are going to split C3. We have C3 here, and they become C3A and C3B. So you split C3 by binding those 3 support molecules. The last one, the most recently found, is what is known as the lectin pathway. And the more research we do, the more we learn about lectin and microorganisms. Here, I'll draw it under my interferon drawing. So here, we have our pathogen. There's our big old bacteria. And bacterial cells have a lot of mannose on them. It has a sugar as part of its cell wall. And so what are known as mannose binding lectins, which is also some of these support proteins, are also going to split C3. C3B, not 3,3. Once you know the 3 pathways that activate, and all these have to do with basically complement binding the surface of the pathogen. Right? These pathogens tend to be bacteria, by the way. They don't tend to do this on viruses. The other take-home message here, this C1A, C1B, I don't know, have you any? Just try that if you've had cell signaling pathways. Do they do any of that in anatomy, physiology?
Maybe a little bit.
but not much. Yeah, I know 161 we didn't either. So, the way this works, it works like a pathway pattern, whereas C1A and C1B, and I admit I don't even remember which one, they're gonna split C2, which will actually split C3. So what I'm getting at is C3 is the central component of this whole process. Let me get rid of my toll-like receptors, which I drew way too big. So if you look at this as a process, you get C1, which gets split to C1A and C1B, which is going to split, I want to say it's the B one, split C2 to C2A and C2B, which is going to split C3. Right? C3 is going to give us C3A and C3B. And our other pathways, right, they did the same. Our alternative did this, and our lectin pathway did this. So this process, I guess I should draw those arrows more towards C3. Be more like this. But C3 is really the central molecule here. Splitting C3 is what sets this thing off. I don't think your book had a great diagram of that. What I grabbed was just the bottom part. So splitting C3 is what starts the response process. So activation leads to the splitting of C3. Response goes, again, there's 3 different responses, and they all happen. It's not like it's one or the other. Okay? I don't care that you know really the different complements that are involved in these, other than that C3 is like the key. The first response is called opsonization.
So you said C3 triggers the whole response? Is that what you said?
So the triggers are these: the antibodies, the BDP, and the mannose.
Okay.
C3, I guess, is what's going to bring those together to cause the response, if that makes sense. I'm trying to think of a good thing. Just think like turning on a car. You can push a button, you can turn a key, right? I'm sure there are other ways, but they all lead to the car starting. And so that's kind of like the car starting. And then this is what happens after, going forward, going backward, whatever else. In opsonization, C3b binds the pathogen. And it's just a tag. It tells phagocytes, it tells all sorts of things that this is a cell to be destroyed. So opsonization itself, and we'll see this term again, that just means enhancing the destruction of the pathogen. Right, so C3b binds the pathogen, tags it for destruction. Phagocytes can do that, cytotoxic T cells can do that. We'll see this is what antibodies do too. Antibodies provide opsonization. They tag things for destruction. Second thing that happens here is the inflammatory response. Right. Here, C3A and C5B, right? No, C5A. Right. These are going to induce the mast cells. Remember we talked about them that are in our tissues. They secrete histamine, cause the inflammatory response, right? So these induce the mast cells to release histamine. So neither of these are direct killers, right? We're just tagging things to be destroyed. We're bringing in the cells that are going to do the destruction. There is a third response that is a destructive response. It's called the membrane attack complex, or MAC. We're not gonna worry about A's or B's or any of that, but C5 through C9 basically form a hole in the cell membrane and cause it to leak contents. And I'm just gonna say C5 through C9. Really takes all of them interacting, and they make holes in membranes. And we'll see this a lot for the larger parasite infections, that the way to get a parasite, maybe to not kill it, but to make it miserable, is to just poke a ton of little holes in it. If you do enough of those, it's gonna leave, right? Because like a parasitic worm, it's huge. It's bigger than your immune cells, so would you at least have to make it miserable? So again, 3 ways to activate, 3 ways to respond. And there are actually more responses out there, but these are the 3 big ones. I don't know how much they did this in immunology. There are a lot of diseases of the complement system. So a lot of people are lacking one or more of these proteins because of mutations, and it does inhibit immune response. So these are really big at ramping it up. On the flip side, once these are split, they're active, and like, you don't want massive inflammation, right? You don't want systemic inflammation. So our blood constantly has circulating molecules that will break down these activated complements. Okay. Questions on complement? All right, so that's one big topic. So again, definitely you can lack complement proteins, and a lot of people do. Other things that bacteria can do, well, they can have like a sticky coat, like a capsule, and then all of a sudden the complement proteins can't bind the outside of the bacteria. So they can evade complement that way, and if you have a sticky coat like a capsule, you can't make the pores either. And so we're gonna learn capsules are just really good defensive parts of the cell. They can have other surface properties as well. And then there's actually a protease out there that some pathogens release that digests C5a. Well, C5a is part of the inflammatory response, and it's also part of this membrane attack complex. So it'll really reduce complement effect. So microorganisms have definitely developed, and when I say microorganisms, first bacteria, have developed methods to avoid these. Questions? I'm trying to give you how this helps us and how the bacteria avoid it. All right. In that case, let's do the other really big one. I'll erase as I go, I think, which is phagocytosis. Does anyone remember what that word means? Yeah, cells eating cells, or we just call it cell eating. Okay. So we're going to look at the process, because this is really the main killer, right? Neutrophils come in first. They're really, really good phagocytes. They're going to eat whatever gets into that initial site of infection. Neutrophils die really fast. They don't really discard all the waste very well. But by then, the macrophages usually come in, and they're even better phagocytes. And they will eat up everything, and they are good at dumping waste. So they dump waste. They make more digestive enzymes. They keep going. We'll also, after the exam, talk about cytotoxic T cells, which are another type of phagocyte. Actually, they're not phagocytes, but they are killers. There are lots of good phagocytes out there. And these are going to be the big killers. For now, the ones we're going to talk about are the neutrophils, which come in first, those macrophages, which follow, And then we have the dendritic cells. There aren't as many of them, but they grab some pathogen and they take it to the lymphatic system and they introduce it to your adaptive immune response. All right, so we got our dendritic cells. Well, first thing that has to happen is we have to get our phagocytes where they need to be. And so we usually say the first stage of phagocytosis is chemotaxis, right? Just moving toward the area. Ways they are drawn there. The pathogen itself secretes stuff. So the pathogens secrete chemicals. We also had our cytokines. There are multiple cytokines, multiple chemicals of the immune system. Histamine is going to help draw these things in. We're just going to say cytokines and not be too specific. Okay, so cytokines from your immune system and from damaged cells. Cytokines from your cells are also going to bring these guys in. Complement will bring them in, right? C5a here and C3a. So complement will bring them in. So lots of different attractants to bring these guys to the site where they need to be. Questions on that one? Keeping that pretty vague. Okay, once they get there, how do they know what to eat? And I just erased my toll-like receptors. But generally, it's items on the surface of the pathogen, which your book calls these pathogen-associated molecular patterns. We're going to do attachment. And I'm just going to do PAMPs. These patterns are going to be on the outside of the pathogen. That's the take-home message there. It can't be like the DNA that's on the inside. It needs to be on the outside of the pathogen. And so if you're thinking, here goes my brain, like antigens, like something an antibody would bind, is a really good example of a PAMP. And so that's what our immune system or our phagocytes are going to recognize as well. When these bind, cells are going to release a lot of cytokines, both our phagocytes and other local cells. It'll stimulate phagocytes to divide and come. It'll also stimulate the B and T cells. But for now, we're just going with attachment. This is going to stimulate our phagocyte to ingest this thing. Attach to the pathogen, get some cytokines released, and you're going to ingest. Your book says engulfment. I don't know what terms they're going to use. Hopefully you remember amoebas and pseudopods. These do it a little different. They don't make big pseudopods, usually just small extensions. And they're going to bring the pathogen inside the phagocyte in a vesicle. We don't want spam. So, you're going to use phagocytes. You're going to use pseudopods. To bring the pathogen into a vesicle. And this vesicle has a very special name. It's called a phagosome. I know you can't see I guess you can see it on the overhead. Pretty good. Let me try to write it. So it's going to bring it into this phagosome. But it's just a vesicle inside the phagocyte. Well, now you got the pathogen inside. You need to destroy it. Hopefully you remember from Gen Bio or somewhere, the organelles, one of the organelles is a lysosome. And so the phagosome and the lysosomes are going to fuse. And all those digestive enzymes inside the lysosome are going to start digesting the pathogen. So what we have is the phagosome and the lysosome fuse. And being ever so good with words, call this a phagolysosome. Just combine the two words. Phagolysosome. Remember, the lysosome is filled with digestive enzymes, so this is going to digest the pathogen. We're going to digest the pathogen. Well, if it's a neutrophil, kind of stops right here. Doesn't really do much else. But if it's a macrophage, that phagolysosome is going to migrate to the cell membrane and secrete all the remains out into the environment. Right? So destruction, digestion, and then exocytosis. And this is more for your macrophages. You're going to secrete the remains out into the surroundings. I'll give you a little more idea of what happens during digestion, what's all in there. pH drops that activates the enzymes in the lysosome. Lots of enzymes in the lysosome. There's these things called antimicrobial peptides. That's where that lactoferrin is to take up the iron from the pathogen. There's reactive oxygen species. There's all sorts of things that are in there. All right. Destruction, digestion, spit it out, and they're ready to begin again. Neutrophils, the complement of lysosomes they have is set. So you make a neutrophil, make it has a dozen lysosomes, and that's all it's going to have. Macrophages are constantly making more lysosomes, so they can keep going and going and going as needed. Right. Questions on this? I'm going to talk about, this is really the area where pathogens excel at evading and even manipulating our phagocytes. So, certain organisms, especially ones with capsules, again, the phagocyte can't bind. Capsules is that sticky outer coat. It just makes it hard for anything to bind to that organism. M proteins are found on Neisseриas, think gonorrhea, and they also basically make the organism invisible to the phagocytes. We probably won't have time near the end of the semester. These organisms, they're constantly mutating these M proteins, so you can actually tend to get repetitive infections with organisms that have M proteins, which sucks. And biofilms are like capsules, right? There's this big mucousy coating over top, and so again, the phagocytes can't get in there. Other things pathogens do other than just stopping from getting ingested. Some of them will get ingested, and then when they'll get in that phagosome, it merges with the lysosome, and they actually break the phagolysosome open. so that all those enzymes digest the inside of the cell. And if the bacteria survives, then it has a nice hiding place, just lives inside that cell. Some of them will actually grow inside the phagosome, and some of them will lyse the phagosome and grow inside the cell. Tuberculosis is one of the masters at this, where it will actually hide inside of phagocytes, and that's how people can get these infections that last for a very long time, because it just hangs out. It usually makes this big thing in your lungs, and it just hangs out there, hiding inside your own cells in your lungs. Questions on that? Okay. Do one more kind of big thing about our innate immune system. We're not going to do as much with these last parts. Inflammation. Right? You all know when you get a cut, an injury, right, it inflames. When it inflames, we tend to have swelling, called edema, redness, heat, and pain. All four of those symptoms are caused by the movement of blood and white blood cells into the area of inflammation. Okay. We're going to break it into three parts. Part one is vasodilation. That's where the blood leaves the capillaries and enters the tissue. And then part two, phagocytes are going to follow. And then finally, you got to clean it up, which we're not going to talk very much about. We're more interested in one and two in here. It's a micro class. Okay. Again, if you've had A and P, this is probably old news. So, first, cells get damaged, right? For some reason, in every book I've ever had, they love to show someone getting stabbed. They oftentimes will do it in like the foot for some reason, which just to me seems very painful. But you get an injury. And so the cells in that area are going to release a ton of different cytokines. Histamine, kinins, prostaglandins, leukotrienes. They're going to release all these things into the surrounding tissue. And those are going to encourage some things to happen. Okay, so first, cells in the area of damage are going to release some cytokines. Those are going to cause the local capillaries to expand or dilate. Number two. So local capillaries expand. This is known as vasodilation. And so what happens when they expand, they become leaky. The space between the cells of the capillary widen, and blood and cells can get through those holes. Right? So this is going to allow cells and fluid to leak from the capillaries into the damaged tissue. Which again, you're going to get all that good stuff that's floating around in your blood. You're going to get the complement. You're going to get your iron binding proteins. You're going to get some antibodies. They're all going to enter that tissue. Right, so the first thing that's going to enter tissue is just blood and kind of the smaller things, our proteins and our chemicals. Blood and proteins enter the tissue. So it's going to start swelling up, right? It's going to get red and it's going to get hot. As this continues, the walls of the capillaries get sticky and white blood cells start sticking to the walls of the capillaries. This is what's called margination. So we're really thinking here neutrophils and monocytes and our dendritic cells. So in margination, So think about neutrophils. Originally doing neutrophils and monocytes are going to stick to the capillary walls. Maybe I'll just stop writing. I don't know. Right? The next thing they do, all right, compliment, go. Neutrophils are gonna slip through first, and they basically have to kind of flatten themselves to squeeze between the cells, right? This has a really wild name called diapedesis. Did I spell it right? Diapedesis, yeah. So first neutrophils are gonna slip between the walls of the capillary, between the cells there, and move into the tissue. So our neutrophils are gonna go first, and they're gonna squeeze between the cells in the capillary walls. We got 10 minutes, we're good. After the neutrophils comes the monocytes. And the monocytes are kind of wild. Monocytes move next. And as they squeeze, they mature. And so most of these are going to become macrophages. This is why when they're floating around in the blood, they're not active. But once they go through this process, it activates them. We just say they mature. And a few of the cells will become dendritic cells. And that's brought about by that process of diapedesis, of squeezing between those cells. Right? And your book has a nice image here where the neutrophils got through first and began cleaning up the mess. And then a day or two later, the monocytes start moving through, maturing as they do into macrophages and dendritic cells, cleaning up the dead neutrophils and anything that's left behind. And then later on, if this isn't cleaned up, you'll get some B cells and T cells in there, but usually it doesn't last that long. The last part, which we're really not going to talk about, is tissue repair, right? Platelets, clotting factors all have to move in there, seal it up. Mitosis happens, new cells are made, repairs itself. If there's pus, it will get either released or it'll get reabsorbed, and those materials get used. We're not interested in tissue repair here. Okay. I want you to know capillaries are going to dilate or expand, blood and proteins enter first, and then cells start entering, neutrophils will come second, and our macrophages will come last. Questions on that? All right. Then the only really last thing that was on here, one, local acute inflammation is usually good, right? Especially if it's short term. Cleaning up the mess, it's great. Chronic is bad because obviously it's not able to deal with whatever got in there. And of course, systemic inflammation is horrible, right? You don't want this happening throughout your body. The last part of this chapter, and we're not going to do much with it, is fever. So whereas inflammation is hopefully local, fever is going to be systemic. Usually fever happens if it's for an infection purpose, it's because of a bacteria, specifically gram-negative bacteria. The outer membrane, the lipopolysaccharide, actually stimulates fever. A little bit about this, but the hypothalamus is what regulates your body temperature if you haven't had A&P, and so there are chemicals released by the presence of these bacteria that cause the hypothalamus to reset higher. These chemicals, your book is gonna call pyrogens. I just want you to know there are chemicals that stimulate this. But usually you get interleukin-1, so that's another of our cytokines, and that tumor necrosis factor. These are the two cytokines that are gonna reset your hypothalamus. Fever's generally good again. Fever's gonna stimulate your cells to work faster. So monocytes dividing into macrophages, they're gonna divide quicker, right? They're gonna make more lysosomes. Most bacteria don't like it hot, so fever usually also suppresses the bacteria from dividing. So you're enhancing your immune system and suppressing the bacteria. Obviously, you get too high and you're damaging your own cells as well as the bacteria. So mild fevers are good, like 101 to 103. Get up into that 104, 105, and nobody's getting any benefit from that, you or the bacteria. But again, short-term mild fevers are very beneficial. Enhances your cells, inhibits bacterial cells. All right, that was really everything. This is just how all the ways it stimulates your body to do all the things it should do. But again, that's up to about 103. Get much above 103 and everything is shutting down. Questions on that? I don't want to keep hitting you guys with too much more. So, that's where the exam's gonna end. So starting with transcription, and then going through all the ways mutation and horizontal gene exchange, and we'll just do the innate immune system. Right? And when we get done with the exam next Wednesday, we'll talk about the adaptive immune system. I have one more ed puzzle I'm going to put up. I'm hoping it's going to work because it doesn't want to save. But everything else is up. We'll talk about